All right, we're talking about everything membrane this week. We're going to start by discussing the structure, and then in subsequent videos, we'll get into the physiology, basically how the membrane works, and also a little bit about the special case of how water moves in and out of the cell. So here are some objectives, and let's just dive right in. So the cell membrane, or also known as the plasma membrane, is the boundary of the cell that separates it from the environment. You need a way to say this gooey stuff is me and that gooey stuff is not me. So that's where the membrane comes in. It's a boundary. It acts kind of like the bouncer at a club where it controls what goes in and out of the cell. It's highly flexible and fluid. It's a really cool structure. It's about 8 nanometers thick, so pretty small overall. This is an actual electron microscope image of the plasma membrane, which is really cool. And then, as I said, its main function is to control what goes in and out of the cell. So let's take a look at this image, and we're going to start breaking down all the different parts of it and what they do and the characteristics of the cell membrane overall. So number one, the cell membrane is fluid and flexible. What we mean by fluid is not necessarily that it's a liquid, but that it is flexible and it constantly has all of these pieces in motion. So nothing within the membrane itself is ever truly static. Things can move around, they can kind of slide past each other, that kind of thing. It is also selectively permeable. We'll get into this a little bit more later, but basically the function of the membrane, as I said, was to control what goes in and out. That is selective permeability, choosing what passes through and what does not. And this same double layer structure is the same for the plasma membrane itself and for the membrane organelles. So the membrane organelles might have some differences in terms of what kind of membrane proteins they have. They usually won't have glycolipids on them or glycoproteins or anything like that necessarily, but they will have this two-layer phospholipid structure that we're going to review a lot in this video. And the membrane is primarily composed of lipids. They are the number one molecule type. They are the most prevalent molecule. They make up the characteristics of the membrane. And so if we remember back from talking about biomolecules, our lipids are the fats, the waxes, the oils. So what we're discussing here is something highly nonpolar, and that's going to be really, really crucial to how the membrane operates. So on top of it being primarily composed of lipids, even cholesterol is a lipid, we do have some proteins and some carbohydrates that we're going to talk about later. But they are secondary to the lipid portion of the membrane. So the membrane itself is primarily composed of these lipids called phospholipids. And so if we look at that word, they are a lipid, but this lipid has a phosphate group in the head. And the phosphate group is highly, highly polar, but the rest of this molecule is not. So it's these two hydrocarbon tails. What we mean by hydrocarbon is just a chain of carbons with a bunch of hydrogens on them. We discussed this a little bit about the differences between being fully saturated with hydrogens, or maybe you have a double bond so you don't have quite as many hydrogens. That's the same thing we're talking about here. And then it will be connected to this polar phosphate head. So as I said, this phosphate group is highly polar, meaning it is hydrophilic. It's going to be attracted to water. However, these tails they're lipids, so they are nonpolar. They are hydrophobic. As a result, they're going to want to repel water. So we have this really interesting molecule that is amphipathic. Amphi means both, and pathos means experience. So this is a molecule that has the experience of being both a polar and a nonpolar molecule. that's a little weird to think about, but it's going to really inform the function of this particular molecule. Remember, function tells us what the structure tells us what the function is going to be. So when we see a molecule that has a polar component on the opposite side of a nonpolar component, we can kind of get our minds starting to think about how those will come together and how they will function in terms of a membrane. So let's get into that a little bit. These phospholipids will naturally form bilayers. Bi means two, so they will naturally form two layers when they are placed in aqueous solutions, in solutions that are water-based, where water is the solvent, as water we discussed as the universal solvent. So since the tails are nonpolar, they're lipids, they're hydrophobic, they do not want to associate with any water. Well, the exterior of the cell is an aqueous solution. The interior of the cell is an aqueous solution. So their only hope is to face each other and form this double layer where these hydrophobic tails are hidden away from the water. And what's hiding them? Well, these two layers of hydrophilic heads with that phosphate group in there. So these hydrophilic heads want to associate with the water. So it works out best for this molecule. It creates this really waterproof barrier that's very nonpolar on the inside, while still being able to associate with the external aqueous environments and the internal aqueous environments. So it creates a tight barrier to anything that is polar or hydrophilic, because this is a very nonpolar hydrophobic area, so those hydrophilic polar molecules won't want to pass through. So I also mentioned that the membrane is fluid and constantly changing. If the whole goal of the membrane is to control what goes in and out, then things have to be able to get in and to get out. If the membrane was not fluid and it was very static and it was very inflexible, how would we be able to get things in and out? So the phospholipids are constantly moving around each other. Some of the phospholipids, as you can see in this example image, have unsaturated fats. So this one right here represents a saturated fat that is straight. But when we have an unsaturated fat, it creates that bent kink shape. And as a result, it ends up creating a little bit of space. These phospholipids with kinks in them won't line up and stack quite as close together. So having a little bit of space between one phospholipid and another allows them the freedom to have this movement within the membrane itself. So we can see here's that example where this allows it to be a little bit more fluid, but if they were all saturated bonds, they would end up packing together very, very tightly, and this would not be as fluid. It would be more viscous. It would be a little bit more hard, and it would make molecules struggle to get in and out. We also have cholesterol. This is in animal cells. And what it does is it helps to increase the fluidity of the membrane when we are lacking these saturated fatty acids. So overall, we call the membrane a fluid mosaic model. Fluid because we have constant movement, and a mosaic because we're not just made of phospholipids. We have cholesterol, and as we'll see in a minute, we also have proteins and carbohydrates. So a mosaic is a picture that comes together by taking unrelated parts and bringing them together. So usually we think about mosaics in terms of bringing lots of different little tiles together to make a broader picture of like a person's face or something like that. Each tile is different, has a different shape, a different color, but together they come to make a beautiful picture. So that's where we get this idea of the fluid mosaic model. So throughout the membrane, we will have lots of different membrane proteins, and their primary job is to transport those polar or charged molecules that otherwise cannot get across the membrane. So those are our transport proteins. They'll act as like little channels. Some of them can freely move through the membrane itself. Others are anchored in place by the cytoskeleton. But that's just one particular job of membrane proteins. We also have membrane proteins whose job is to hold identity markers, like basically hold the cell's name tag up. We have some whose job is to connect one cell to another or to initiate a chemical reaction. They'll act as like an enzyme inside the membrane. There's lots of different jobs that these membranes can take. And so here's just an example of those many different functions. They can be those channel transporter proteins, they can be enzymes. We will discuss this signal transduction. Basically, if a molecule can't get into the cell, it can sometimes talk to a protein, and that protein can create a signal to propagate that message inside the cell. But we'll get into that more when we talk about the endocrine system and hormones. It can carry those identity markers, we can have cells adhere to each other, and we can anchor certain ones in place with the cytoskeleton. Now, the last piece of this mosaic are the carbohydrates. So on the membrane, you will often see carbohydrates either attached to the head of a phospholipid or to a protein itself. And these actually end up giving our plasma membrane, the overall cell membrane, an inner and an outer face, because we only see these carbohydrates form on the exterior surface of the cell membrane. They do not form on that interior surface. So I like to remember it by thinking, oh, the cell is candy coated. We have sugars that only coat the outside of every cell. So we primarily use these for cell-to-cell communication and cell recognition. We often call some of these antigens. If you've ever heard of an antigen, basically it's a marker that tells the immune system, hey, this is our cell, or hey, this is not our cell, and you got to attack it. So antigens are just chemical markers that help the cells distinguish self from non-self. So that is it for this video. There are going to be 2 more, so make sure you review those as well.